one of my students was trying to do this kind of experiments by mixing these cells with the cancer cells and also the T cells. That he showed me that before the co-culture, actually the tumor cells were growing in a monolayer. But upon contact, you can see for a certain period of time after the co-culture, the tumor cells, the much bigger cells you can see here, actually try to clump together and change their morphology while they're being surrounded by the arrows that we cannot see, the much smaller lymphocytes that has been mixed in the co-culture. We do not know why such we do not know why the tumor cells will have such kind of change in morphology, and it's also not present in all the cell lines. That this will happen on exposure to T cells. And so we're trying to see what was actually happening with further co-culture of the different tumor cells with the T cells isolated from patients. And it seems that if we measure the PD-L1 expression on exposure to T cells, it seems that most of them will give certain response of PD-L1 expression in the tumor cells after exposure to T cells. And the T cells having finite lifespan in the culture usually, again, will give some increase in the expression of corresponding PD-1 on the surface, but this will actually this will actually drop to a certain level after they have done the battle to try to kill the tumor cells. We are still exploring further on how to explain these, and it seems that the T cells are effective in their killing for a certain moment after the co-culture with the tumor cells. But this cytotoxicity will drop after prolonged co-culture. So it seems to us that the T cells, they can do the job. They can try to kill the tumor cells, especially if the tumor cells express PD-L1. But there is a dynamic mechanism underneath that the tumor cells may change their expression, and the T cells need some help in acting on the tumor cells in order to maintain the killing. So that appears to imply to us that these kind of immune checkpoint inhibitor could be effective, but at the same time needs further development and needs further research into better preservation of cytotoxic T cell killing for the patients. And we are trying to analyze the different proteomic profile and gene expression to see if we can detect the relevant dynamic changes of these tumor cells and T cells after co-culture in order for us to better understand what is the exact battle that is going on between the tumor cells and the T cells and how to augment that for better therapy in the future. After talking so much about these different markers, EGFR, ALK, and also PD-L1, can we detect them just in the tumors? Now, a lot of the times in the clinical situation, we have difficulty in getting good going biopsy because the lungs are deeply seated organ, and the tumors, a lot of the times, is also deeply seated. So it would be good if we can perform liquid biopsy with trying to detect these in the blood. And it depends on whether we have the available specimen, whether we have appropriate method for the isolation and detection of these biomarkers. And you can see, in comparison to of the circulating tumor cells with the tissue diagnostics, of course, we can detect in either specimen. But for tumor biopsy, a lot of the times, it allows diagnosis and rebiopsy and need for rebiopsy. But for the circulating biomarkers or liquid biopsy, there is a great advantage here is that we can try to perform serial monitoring for our patient instead of going for invasive investigation every time you want a monitoring or for biopsy of the patient. The requirement is that you can detect it from the circulation or even in the body fluid. So the aim would be to try to take a blood sample or a biospecimen sample, and we can try to use that sample, the biomarkers detected in that sample, for diagnosis, to guide our therapy choice, and to monitor the disease progress if possible with liquid biopsy, and also, if possible, to try to identify new targets or for early detection of disease recurrence if possible. And of course, if present, we can use it for prognostication also. And there are now panels of oncogenic driver mutations that can be detected in commercially available platform for detection of these, all the different genes in a blood sample. Of course, it will give rise to a better understanding of the biology of the tumors, but at the same time, for an individual patient, you may be buying things that you don't use. So a lot of the times, we will try to see if we can detect specific genes or mutations in one patient, one at a time. So take EGFR mutation as an example, and is now available for detection in the blood. But it will depend really on whether you have a test that is specific enough to pick up the EGFR mutation in the circulating tumor DNA in the blood compared to the normal DNA that is present in the blood. So the mutation is present in only a minute proportion of tumor DNA present in the circulation among the presence of normal DNA. So you need a very specific technique in order to amplify the minute amount of circulating tumor DNA. And this is already available with digital PCR being the best technique for picking up these minute amounts of DNA. We also have developed this array method that initially to try to detect all the different kinds of EGFR mutation and of relatively good performance. And it has been also shown in our panel that if the tumor DNA is actually present and detectable in the circulation, remember, not all of them can be detected. It will depend on whether the tumors will leak that circulating tumor DNA into the circulation for you to pick up in the circulation. So it has been shown before that if the tumor DNA is actually leaked and be detected in the circulation, it implies a relatively poorer prognosis for those patients. So the high levels of circulating DNA actually correlates with the prognosis, but it really depends on the technique that will be able to detect those minute amounts of circulating DNA in the circulation. We are following up patients with serial profile to check if we can use it to detect the disease recurrence with the resurgence of the mutation copy number in the circulation after the patient has got responded for long period after treatment with EGFR TKI. And nowadays we have different methods of detecting circulating tumor materials, and you can detect tumor the circulating tumor cells, which are actually most of the time 1 or 2 cells among the sample that you can pick up in the patient's blood sample. We have mentioned about circulating tumor DNA, but at the same time, we are also trying to explore newer markers like the exosome that I tried to introduce to you earlier in the ALK story, for the exosomal microRNA. And lots of the times, we know that these exosomal